Are you ready for a wake-up call about your diet? A recent study has uncovered a concerning link between the consumption of ultra-processed foods and significant changes at a molecular level, potentially impacting our health in ways we're only beginning to understand. This research suggests that what we eat could be leaving a lasting mark on our bodies, right down to our DNA. This intriguing research, published in the journal Nutrients, delved into how ultra-processed foods, UPFs, affect our genes. The study focused on DNA methylation, a crucial process that influences how our genes behave. So, what exactly are ultra-processed foods? According to the NOVA classification system, UPFs include those convenient, often tasty, and long-lasting foods like ready-made meals, packaged snacks, and sugary drinks. They're packed with preservatives, flavorings, colorings, and other additives. While convenient, they are often energy-dense but low in essential nutrients, and high in unhealthy fats, sugar, and salt. But here's where it gets controversial. The study found that women who consumed more UPFs showed differences in DNA methylation compared to those who consumed less. Specifically, the high UPF group had 80 differentially methylated regions, DMRs. This suggests a potential epigenetic link between UPF intake and health outcomes. The researchers examined a group of 30 women aged between 20 and 40 years old. Their diets were assessed using food records, and they were divided into groups based on their UPF consumption. The high UPF group consumed an average of 45% of their total energy from UPFs, while the low UPF group consumed only 14%. And this is the part most people miss. The study found that most of the DMRs were hypomethylated in the high UPF group, meaning that higher UPF intake was associated with lower DNA methylation levels. The study highlighted 7 regions with the largest methylation differences, including RNA5S7, RNA5S9, RNA5S13, link 00396, FOXP1 LOC124902961, and REPIN1 What does this all mean? The study suggests that UPF consumption may influence gene regulation through epigenetic modifications. Some of the affected genes are involved in metabolic regulation and cancer-related pathways, hinting at potential links between UPF intake and adverse health effects. However, it's important to note the limitations. The study was a pilot study with a small sample size, which limits the ability to draw definitive conclusions. The cross-sectional design means that it cannot prove cause and effect. The study also acknowledges that the results are exploratory and need further investigation. The study's findings are a significant step forward, highlighting the potential impact of UPFs on our health. It underscores the importance of a balanced diet and encourages further research into the long-term effects of UPF consumption. What are your thoughts? Do you think this study raises valid concerns about the impact of ultra-processed foods on our health? Share your opinions in the comments below.